السلام عليكم. معكم دانا أبو بكر من كلية طب الأسنان. إن شاء الله اليوم رح نتكلم عن رح نكمل على Digestive Tract. رح نبلش Small Intestine and ending with the Large Intestine. So the small intestine is the site where digestive processes are completed. احنا بنعرف إنه في عنا incomplete digestion بالoral cavity بالstomach. The small intestine is responsible for completing the digestion and it's specialized in absorption. بصير في عنا absorption of nutrients اللي هما the products of digestion through the epithelial lining of the small intestine. The small intestine is relatively long, it's about 5 meters in length and it's the longest part of the GI tract. It consists of 3 segments, the duodenum, jejunum and the ileum, and these segments have most histologic features in common and they are discussed together. So the mucosa, once it's viewed macroscopically, you can see a structure called plicae circularis or circular folds. هذول plicae circularis are permanent circular or semilunar folds, يعني they're either circular or semilunar. And they circulate the small intestine, as you can see here. Okay, they are made up of mucosa and submucosa. They are mostly seen in the jejunum and they increase the surface area about 3-fold. Okay, they are a series of permanent circular or semilunar folds نفس ما حكينا هلا. They consist of mucosa and submucosa and they are best developed in the jejunum. عندنا كمان الvilli. The villi densely cover the entire mucosa of the small intestine. They are short mucosal outgrowths. They have a length of about 0.5 to 1.5 millimeter and they project into the lumen. They are finger-like or leaf-like projections. They're covered by a simple columnar epithelium of absorptive cells called enterocytes. وبين الانterocytes عندنا goblet cells that are interspersed. Each villus has a core of loose connective tissue extending from the lamina propria and it contains fibroblasts, smooth muscle fibers, lymphocytes, plasma cells, fenestrated capillaries and a central lymphatic called a lacteal. So عندنا plicae circularis responsible for increasing the surface area. عندنا كمان villi which is also responsible for increasing the surface area. The villi projects into the lumen. It is a mucosal outgrowth. It's short. It has a finger-like or leaf-like a structure. And it's covered by a simple columnar epithelium of absorptive cells called enterocytes with goblet cells interspersed between them. They have a core of loose connective tissue extending from the lamina propria, containing fibroblasts, smooth muscle fibers, and so on. Between the villi are openings of the short tubular glands called intestinal glands or crypts of Lieberkuchen, and these crypts of Lieberkuchen are invaginations of the surface of the epithelium into the lamina propria. The upper part of these crypts is occupied by enterocytes and goblet cells, and the lower part is occupied by DNES cells, Paneth cells, and stem cells. And the epithelium of each villus is continuous with that of the intervening glands, and يعني the epithelium of the villi is continuous مع الepithelium of the intervening glands. And the epithelium of the intestinal glands includes differentiating cells, pluripotent stem cells for all the cell types of the small intestine. يعني في stem cells that are ready to replace all the cell types بالsmall intestine. هدول cell types رح نتكلم عنهم هلا, starting with enterocytes. So enterocytes are the absorptive cells of the small intestine. They are the most common cells. أكتر شي موجودين, they're the most numerous. And they are tall columnar cells. They have an oval nucleus that's located basally. The apical end of each enterocyte displays a prominent ordered region called the striated or the brush border. هاي brush border is seen to be a layer of densely packed microvilli covered by glycocalyx. So I just want to find the picture to show you what I mean. So hold on. هاي مثلا زي هون, as you can see, this is a brush border. As you can see, they're densely packed microvilli. And these microvilli are covered by glycocalyx نفس ما حكينا, and the glycocalyx is responsible for increasing the surface area even more. They have adhesive properties and they are responsible for the intake of nutrients into the cells. The microvillus is a cylindrical protrusion of the apical cytoplasm and it's about 1 micrometer tall and 0.1 micrometer in diameter. They contain actin filaments and they're enclosed by a cell membrane. Each enterocyte has an average of 3,000 microvilli and each 1 millimeter square of mucosal surface area of the mucosal surface contains about 200 millions of these structures. So as you can see, they greatly cover the mucosa and the small intestine in general, and they increase the surface area greatly. وعندنا هدول structures اللي هم المايكروفيلاي الفيلاي and the placocercularis because the small intestine is specialized in absorption. ف we need these structures عشان يصير في عنا a more efficient absorption لأن كل ما زاد ال surface area كل ما زاد ال absorption and كل ما زاد ال chance of an efficient absorption. So as we said هلا it's an important feature in an organ specialized for nutrient absorption, and it is estimated that the placocercularis increases the intestinal surface area threefold, the villi increases it tenfold, and the microvilli increases it another 20-fold, resulting in a total absorptive area of over 200 meters square in the small intestine. So the small intestine is 5 meters in length, approximately 5 meters in length, but it has an absorptive area of over 200 meters square in the small intestine, and this is because نفس ما حكينا هلا لازم يكون في عنا كتير surface area عشان يصير في عنا a more efficient absorption. And just a quick note is the villi is much frequent and much taller in the duodenum. The mechanism of nutrient absorption varies with the different molecules produced by digestion, so each molecule has its own way of absorption. مثلا disaccharides and peptidases, which are secreted by the enterocytes and located within the glycocalyx, hydrolyze the disaccharides and dipeptides into monosaccharides and amino acids. So disaccharides and dipeptides بصير فيهم hydrolysis by the disaccharides and peptidases. بصير في عنا monosaccharides and amino acids عشان يصير في عنا an easier absorption which happens by active transport, and then بيصير في عنا an immediate release again من الانتروسايت عشان يصير في عنا uptake by the capillaries عشان يصير في عنا absorption من البودي. And the fats, their digestion happens by the gastric and pancreatic lipases, which produces lipid subunits اللي هم glycerol, monoglycerides and fatty acids. These subunits become emulsified by the bile salts into small micelles. Micelles are lipid droplets. They're about 2 nanometers in length. And بصير بعدين إنه lipids they enter the enterocytes by passive diffusion. إحنا حكينا هون ال disaccharides and dipeptides they بصير فيهم absorption by active transport. هون by passive diffusion and membrane transporters, and these lipids are then reesterified into triglycerides in the enterocyte smooth endoplasmic reticulum, and then they are complexed with apoproteins in the Golgi apparatus producing chylomicrons. هذول الchylomicrons are discharged from the cell's basolateral surface for uptake by the lacteal, and this is how يعني the fats are absorbed. The doctor didn't mention it at all, but يعني موجودة بالكتاب so it's included. هلا رح نتكلم عن الجوبلت سيلز. so the goblet cells ابدا ما في منهم بالستمك، موجودين بالسمول intestine بالسمول and large intestine and they increase as you go. so بالدودنم ما في كتير، بيصير في عنا اكتر بالجيجنم، اكتر بالإليوم، اكتر بالسمول intestine and so on. They are interspersed among the absorptive enterocytes. هلا احنا حكينا انه they increase as you go, but they are never more than enterocytes. الانterocytes as we said انه they're the most common cell بالسمول بالintestine. the goblet cells are never more than the enterocytes، بس في عنا كتير منهم كمان as we go يعني down the intestine. They secrete glycoprotein mucin, which is then hydrated to form mucus, and the mucus here is the main function of the mucus is to protect and lubricate the lining of the intestine. The Paneth cells are located in the basal portion of the intestinal crypts, below the stem cells. They are exocrine cells with a large eosinophilic secretory granules in their apical cytoplasm. The Paneth cell granules release lysozymes, phospholipase A2 and hydrophobic peptides called defensins. They defend the small intestine by binding and breaking down the membranes of microorganisms and bacterial cell walls, and they have an important role in innate immunity, and they also regulate the microenvironment of the intestinal crypts. They maintain the intestinal flora by يعني breaking down the cell wall of any foreign bacteria. في عنا هون صورة I believe of the Paneth cells which show the as you can see here they have this is the Paneth cell. It is pyramidal in shape. As you can see هون عنا the eosinophilic part and the basophilic part which stains يعني the eosinophilic part stains red or pink and the basophilic part stains blue. So I just wanted to show you it's pyramidal and an eosinophilic upper part يعني an eosinophilic apical part and a basophilic basal part of the Paneth cell. And they have defensins which defend the small intestine by binding and breaking down the membranes, by breaking down membranes of microorganisms and bacterial cell walls, and they also maintain intestinal flora. So the anterior endocrine cells are present in varying numbers throughout the length of the small intestine. They secrete various peptide hormones. Many of these are of the open type اللي حكينا عنه بالمحاضرة اللي فاتت, in which the constricted apical end of the cell contacts the intestinal lumen, and it has chemoreceptors similar to those of the taste bud. These chemoreceptors sample the levels of certain nutrients such as sugars to regulate hormone released basally. So إذا زاد الsugar they بيصير في يعني high stimulus and they release insulin and so on. So M cells or the microfold cells are unique epithelial cells specialized for transepithelial transport of particles and microorganisms. So the M cells are specialized in transepithelial transport, and they are located mainly in the ileum's mucosa, overlying the lymphoid molecules of Peyer's patches. Each M cell has a pocket formed by the deeply invaginated basolateral membrane containing lymphocytes and macrophages. These lymphocytes and macrophages are covered by a thin layer of cytoplasm. Continuous sampling of the intestinal microorganisms by the M cells and the immune cells within and below the M cell is an important part of the defense provided by the mucosal immunity. So, recap, M cells are specialized in transepithelial transport. They are located mainly in the ileum's mucosa, and they overlie the lymphoid follicles of Peyer patches. Each M cell has a pocket formed by the deeply invaginated basolateral membrane, and these pockets contain lymphocytes and macrophages. And M cells بيصير فيهم continuous sampling of the intestinal microorganisms and the immune cells within and below the M cells. And this is an important part of the defense provided by the mucosal immunity. So the M cells lack microvilli. They have a smooth surface except for a few folds. They're covered نفس ما حكينا the lymphocytes and the macrophages اللي هم بيكونوا موجودين بالpockets of the M cells. They are covered by a thin layer of cytoplasm. And these M cells initiate human response by delivering antigens to the cells of the immunity system. And they also monitor the environment of the lumen by sampling نفس ما حكينا هلا. So other layers of the small intestine هلا رح نحكي عنهم. So along the entire small intestine في عنا loose connective tissue of the mucosal lamina propria. The lamina propria of the small intestine contains extensive blood and lymph microvasculature. It also contains nerve fibers, smooth muscle cells, and diffuse lymphoid tissue. The lamina propria penetrates the core of each intestinal villus, bringing with it microvasculature, lymphatics, and nerves. Smooth muscle fibers extending from the muscularis mucosa produce rhythmic movements of the villi that increase the absorption efficiency. So the microvilli فيها movements, rhythmic movements, and this increases the efficiency of absorption. Fibers of muscularis mucosae also produce local movements of the plicae circularis that help propel lymph from the lacteals into submucosal and mesenteric lymphatics. So البلايكي circularis بيصير في عنده local movements by the muscularis mucosa. والmuscularis mucosa also produce rhythmic movements of the villi, increasing the absorption efficiency. The submucosa of the small intestine has larger blood and lymph vessels, and the diffuse interconnected neurons of the submucosa اللي هم الميتر nerve plexus. The proximal part of the duodenum has in the submucosa and mucosa large clusters of branched tubular mucus glands called the duodenal or the Brenner glands with small excretory ducts. So على proximal part of the duodenum عنا بالsubmucosa and mucosa large clusters of branched tubular mucus glands اسمهم the duodenal glands, or الدكتور اليوم كان بيحكي عنهم Brenner glands مو اليوم on Thursday يعني, with small excretory ducts opening among the intestinal crypts. The mucus from these glands is distinctly alkaline, which neutralizes the chyme entering the duodenum لأن الكايم بيكون جاي من السtomach. So بيكون highly acidic نفس pH of the stomach. So the secretion اللي بتيجي من الducts the excretion اللي بتيجي من الducts, it is distinctly alkaline to neutralize the pH of the chyme entering the duodenum from the pylorus, protecting the mucous membrane and bringing the intestinal contents to the optimum pH for absorption. So the mucus that's secreted اللي حكينا عنه هلا, الميوكس اللي بيجي من البرينر glands is rich in bicarbonate and the epidermal growth factor, and the secretion is stimulated by the parasympathetic system. So لازم يكونوا بالoptimum temperature عشان يصير في pancreatic enzyme action, عشان يصير في عنا absorption and so on. In the ileum, both the lamina propria and the submucosa contain well-developed mucosa-associated lymphoid tissue اللي هو MALT. So the MALT is the mucosa-associated lymphoid tissue, and these consist of large lymphoid nodule aggregates known as the peripatches underlying the epithelial M cells. The muscularis of the small intestine is well-developed, and it's composed of the internal circular layer and external longitudinal layer, and between them في عنا neurons of the myenteric nerve plexus اللي هو Auerbach nerve plexus, which produces peristalsis, and the neurons of both submucosal and myenteric plexus myenteric plexus are largely autonomic and collectively make up the large enteric nervous system. The small intestine is covered by a thin serosa with the mesothelium continuous with that of the mesenteries. So the small intestine نحكي نعرف إنه it's covered by serosa that's continuous with that has a mesothelium that's continuous with that of the mesenteries. So okay, I just want to check إنه ما في أي صورة that we missed. These are important. As you can see هون عنا goblet cells, enterocytes. As you can see في enterocytes that are interspersed between the goblet cells. Same thing here, هي enterocytes في عنا a goblet cell that's interspersed between these enterocytes. And هون في عنا the plaque circularis encircling the small intestine, and they are best developed in the jejunum نفس ما حكينا. هاي الفيلاي. okay, هاي المسكولاريس هون في عنده the inner circular layer and this outer longitudinal layer نفس ما بنعرف. okay, وهون هاي البrush border that's a layer of densely packed microvilli. هون في عنا the intestinal villi. As you can see the goblet cells هيهم. okay, وحكينا إنه they increase down the small intestine and the large intestine, and yeah we discussed this. هون as you can see عنا الانتروسايت موجودة بال upper part of the intestinal gland, وهون عنا البانث cells as you can see here بال lower part, and في عنا كمان an important structure is the venule and lymph vessels and arterioles obviously, and عنا كتير منهم هون because they are important for absorption of the materials from the lumen of the small intestine into the blood. And yes, I believe that's the small intestine. هلا خلينا ننتقل لل large intestine. So the large intestine is the large intestine or bowel is specialized in the absorption of water and electrolytes, and it forms the indigestible material into feces. It has the following regions اللي هما الcecum, ileocecal valve, and the appendix. It also has the ascending, transverse, and descending, and sigmoid colon, and the rectum. The appendix is the lumen of the appendix is usually filled with debris. The mucosa of the appendix has simple columnar epithelium. It has crypts that have different types of cells. It also has a lamina propria that has a lot of lymphoid follicles and shallow crypts. والدكتور حكى إنه the other layers are insignificant. The rectum is where feces are stored prior to evacuation. The mucosa of the large intestine lacks villi, and except in the rectum has no major folds. So the mucosa of the large intestine ما فيها villi, and except in the rectum it has no major folds. So the large intestine ما فيها major folds except for in the rectum. Less than 1/3 as long as the small intestine. The large intestine has a greater diameter, about 6 to 7 centimeters. The wall of the colon is puckered into a series of large sacs called hostra. هلا بنشوفهم. هاي الهوسترا as you can see they're circulations يعني زي هيك. هاي appendix rectum as you can see هيو it's where the feces are stored prior to evacuation بيصير through the anal canal. هاي هون عنا ascending colon, هاي transverse colon, هاي descending colon, and هاي sigmoid colon. We're gonna discuss this all in a minute. عنا كمان the mucosa of the large bowel is penetrated throughout its length by tubular intestinal glands, and these and the intestinal lumen is lined by goblet and absorptive cells. The absorptive cells of the large intestine is called colonocytes, whereas the absorptive cells of the small intestine is called enterocytes. These and the intestinal lumen are lined by goblet and absorptive cells with a small number of enteroendocrine cells. So the lining can بالsmall intestine goblet and absorptive cells only. بالlarge intestine في عنا goblet and absorptive cells and enteroendocrine cells, but a small number of enteroendocrine cells. The columnar absorptive cells or the colonocytes have irregular microvilli and dilated intercellular spaces indicating active fluid absorption. Goblet cells producing the lubricating mucus become more numerous along the length of the colon and in the rectum. Epithelial stem cells are located in the bottom third of each gland اللي موجودة large intestine. So the goblet cells موجودة أكتر شيء بالركتم لأنه إحنا حكينا إنه as you go it increases. بالركتم موجودة أكتر إشي. The lamina propria is rich in lymphoid cells and in lymphoid nodules that frequently extend into the submucosa. The richness in MALT is related to large bacterial population of the large intestine, and the appendix has little or no absorptive function but is a significant component of MALT. The appendix actually has no significance in the body. يعني هاي الزايدة اللي احنا نعرفها I think اسمها الزايدة الدودية. And استئصالها is easy وما فيها أي ضرر once it's removed من the body. The muscularis of the colon has longitudinal and circular layers but differs from that of the small intestine. Muscularis فيها fibers of the outer layer gathered in 3 separate longitudinal bands اللي هما Tinea coli, and the intraperitoneal portions of the colon are covered by serosa, which is characterized by small pendulous protuberances of adipose tissue. هلا رح نشوف adipose tissue. هيهم هدول adipose tissue. As you can see, they cover the large intestine. هي Tinea coli. Tinea coli is actually responsible for the hostra or the circulation اللي بتكون موجودة على large intestine, and the Tinea coli is actually shorter than the large intestine, which is why في عنا هدول circulations or the hostra. Okay, so the distal end of the GI tract is the anal canal. The anal canal is about 3 to 4 centimeters long. At the rectoanal junction في عنا simple columnar mucosal lining of the rectum. بيصير في an abrupt replacement بيصير على طول stratified squamous epithelium. تتذكروا كيف إنه بالesophageal junction في عنا من stratified squamous epithelium لsimpler columnar the simpler columnar epithelium. هون برجع stratified squamous epithelium ففي عنا an abrupt change. هلا رح نشوف هون I believe لا hold on هي الركتم. هون الجنكشن as you can see it's an abrupt change من simple columnar ل stratified squamous epithelium. هاي الانال canal as you can see هاي change هاي replacement يعني. And the mucosa and the submucosa of the anal canal form several longitudinal folds اللي هما anal columns in which the lamina propria and submucosa include sinuses of the rectal venous plexus. Near the anus, the circular layer of the rectum's muscularis form the internal anal sphincter. The internal anal sphincter is involuntary, whereas the external anal sphincter is a voluntary muscle. And هدول الاثنين those sphincters both are obviously contracted at rest, but upon will you can contract you can يعني بيصير في contraction بالexternal anal sphincter even more عشان يصير في عنا excretion. So I just want to show you هاي الanal canal. As you can see هاي الrectum which extends into the anus. هون عنا internal anal sphincter which is the involuntary muscle. هاي external anal sphincter which is the voluntary muscle. They're both composed of smooth muscle. وبهيك بنكون انهينا chapter 15 digestive tract. إذا في أي شيء مو واضح feel free to message me on messenger بأي وقت أنا I'll help. And رح ننزل ملخص إن شاء الله which will contain all the things that we have to differentiate between إنه كله محفظ all the layers, all the structures and so on. And thank you for watching and good luck.